Hi everyone, I'm Ercument Cicek. I'm a main fellow in computation biology at Carnegie Mellon. And today I'm going to present to you our joint work with Catherine Roeder and Begin Soyolu, named MIRA, Mutual Information-Based Reporter Algorithm for Metabolic Networks. And this work mainly done during my PhD. So here I am showing you the basic information flow through the omics cascade. Basically, you have the genome at the very end, which dictates which RNA to be transcribed, and then RNA is translated into proteins. Then these proteins are used as enzymes to convert these small molecules metabolized into each other. And then you observe the phenotype. So the nice thing about the metabolome and the metabolic network is that it's the closest to the phenotype in the omics hierarchy, and it's less prone to regulation. So any changes in the genome or any environmental perturbations lead to changes in the steady state of the organism. So any such changes are exerted through transcriptional changes to reinstate the steady state. So the question that we are trying to find an answer for is, can I use the gene expression levels to understand the stress levels on the metabolic network? So can I go from the array into the metabolic network? So we are talking about the systematic integration of the transcriptome with the metabolome and the metabolic network, and I'm going to try to give you some reasons why we should do it. So the first reason is the metabolome is the closest to the phenotype, as I just said, and so it gives you a great deal of information about the phenotype. Second is the gene expression datasets are quite abundant, so in the GEO database. With the rise of the RNA-seq, you have unsupervised information about the gene levels, unlike the arrays, and unlike RNA-seq, metabolic profiling is usually targeted. So you are targeting a small subset of metabolites that you are interested in. In addition to that, mass spectrometry cannot detect each and every single metabolite. Some metabolites are really volatile, so they are quickly converted into one another, and you cannot detect them. Even if you can detect them, some of the metabolites are not found in the metabolite database, so you might not be able to identify them. And luckily, the reconstructed genome scale metabolic networks are very well defined and annotated with the genes. So there are also some challenges, of course. One challenge might be each enzyme is connected to many metabolites, so it might be hard to distinguish the effects on which parts of the metabolic network is affected. And if you are trying to do something like flux balance analysis, the gene levels do not correlate well with the fluxes of the metabolic reactions, possibly due to regulation. So I have already introduced you to the problem. Next, I'm going to talk about the current approach, which is the reporter algorithm. Then I'm going to talk about our solution, which is a combinatorial and mutual information-based solution. Then I'm going to show you some results on an E. coli gene expression dataset and an autism spectrum disorder gene expression dataset. Then I'm going to conclude. So the problem and the idea is not new. The reporter algorithm has been proposed for this purpose. The main idea behind the algorithm is to use the metabolic network topology to understand the transcriptional regulation. So the algorithm assigns the importance of each metabolite based on the average transcriptional response around the metabolite and the neighboring reactions. So as you see the formula over here, the ZNI is assigned to a neighboring reaction. It's the z-score of a transcription change of the reaction around this metabolite. So it basically averages out all the statistical changes around the metabolite and assigns a z-score to this metabolite. So let me illustrate with an example. So I'm showing you here the transportation of the metabolite coprogen in the middle from the extracellular space into the periplasm and then the cytoplasm in E. coli. So you are seeing the rectangles as reactions, metabolic reactions, and the pentagons, although they are not seen quite well, pentagons are the genes associated with those reactions. So the algorithm first finds the z-score of the change in each gene, and then through a max function assigns a z-score to each reaction, and then aggregates these z-scores to find the score for the metabolite itself. So there are some possible shortcomings with this method. The first is small sample sizes might lead to missing some significant changes. So for instance, in this case, I had a handful of cases and maybe a handful of controls for this data set. So using statistical tests might lead to missing some changes. And the second is z-score aggregation might cancel out some significant changes. Because as I show you over here, the z-score changes from -infinity to +infinity. And -infinity representing no significant change at all, while +infinity is the most significant change. So if you have two reactions like here, and one changed substantially, one did not, adding out those z-scores would cancel out the effects of each other. So I would like to use some scoring scheme which doesn't change, which doesn't affect the result of another reaction. And furthermore, the gene-by-gene analysis might lead to missing some synergistic patterns, like analyzing each gene, finding a z-score, and then through a max function or another aggregation function, assigning a score to z-score to each reaction might miss such patterns between genes. Here is what we are trying to do with MIRA here. So first I want to analyze a combination of genes at the same time. So this information comes from the metabolic network, and I know these genes are associated with a single reaction. So I want to assign a score to the changes of these genes together at the same time. Second, I want to use the mutual information to measure the transcription changes for two reasons. First, it ranges between 0 to +infinity, so it's not going to cancel out any changes. If there is no change, it's going to be 0 if they are independent, so that I'm not going to affect anything else. And it's going to perform with small sample sizes, hopefully, because I'm not using any statistical tests here. I'm using information theory. Let me talk a little bit about the mutual information. So given two random variables, it measures how much knowing one variable reduces the uncertainty about the other one. So in our case, we have two variables. X is the class variable being case or control, and Y is the combination of genes and their expression levels. Let's assume I have discretized the expression levels using a function or something. So in this simple example over here, I have just two levels, a gene being up or down. So my X variable will be case or control, and my Y variable will be the first gene being up, second gene being down, first gene is down, second is down, and so on. So 4 possible values. So if information between these two variables is 0, it means these two variables are independent, which means knowing the expression levels of these two genes give me no further information about the phenotype. As you see the formula of the mutual information, if the joint probability distribution overcomes the marginal probability distributions, the logarithm will be higher, so we are going to have further information. If they are independent, the logarithm is just going to cancel out the rest of it. So I'm talking about discretizing gene expression values up or down, but let me show you how we really do it. So we are using a B-spline based discretization method. So in this figure you see the x-axis is the observed expression levels for gene X, let's say. So the one way to do it will be just draw a line at 0.5, whatever is less than 0.5 is down, whatever is more, it's just up. But this is a deterministic discretization method, and it might lead to it might be just crude. So what we want to do is we want to assign observations into different bins probabilistically. So I want to say it's 0.6 probability up, 0.4 probability down. The way we do it is we define B-spline curves over the domain of gene X. Here you see 6 curves, and each represent a bin. And the y-axis here is the probability of assigning an observation into a bin. So if I observed an observation at 0.5, I am going to assign this observation with less than 0.5 probability to bins 3 and 4, those are the curves 3 and 4, and with less than 0.05 probability to bins 2 and 5. So that's how we discretize the gene expression. If I have an observation around 1, I am almost going to be sure it's going to be in the highest bin, maybe just with a I'm sorry, maybe with just some small probability it's gonna be in the 50. Covered. So okay, I've been talking about bits and pieces about the algorithm. Let me show you the big picture. So the algorithm first starts on the top left, getting the SPML document of the metabolic network, and for each metabolite it extracts the metabolic reactions around them and the genes that are associated with these reactions. So you see groups of genes associated with the blue rectangles, which are the metabolic reactions. Then it gets the gene expression data set. It discretizes the observations with these splines, and then it finds the mutual information between the groups of genes and their expression levels and the class variable. Finally, for each reaction it has a mutual information. Then it averages the mutual information assigned to each reaction to find the value for each metabolite. So then it reports the significant ones as reporter metabolites. Okay, let me show you some results. So the first data set we are analyzing is the E. coli gene expression analysis done by Coward et al. In 2004. So it has 6 knockouts of key transcription regulators of the oxygen response. So we are going to focus on the delta AFNR gene over there. And we are using the IJR904 reconstructed metabolic network. So on the right I'm showing you a basic sketch of the anaerobic respiration in E. coli. On the top you see the outer membrane. Here you see the periplasm underneath, and underneath you see the inner membrane and then the cytoplasm down. The way anaerobic respiration works is, so when there is no oxygen in the environment, E. coli uses dehydrogenases and electron donors like G3P, formate, and so on, to get electrons and transfer them over to the dehydrogenases on the right. And so as these electrons are transferred, hydrogen atoms are pumped into the periplasm, and then in the downstream they are transported back to synthesize ATP. So you see the FNR gene in the pentagon in the very bottom. This is a key regulator of the switch between anaerobic and aerobic respiration. So we are focusing on the knockout of this gene. And on the left, you see in the blue the reporter metabolites reported by our algorithm. So when there is no oxygen, FNR gene is active, and when there is no oxygen, it's inactive. And these changes coupled with the metabolites called glutareduxin you see in the very bottom, and thyroreduxin. So our algorithm finds those metabolites in the reporter set. This gene, the resulting protein, activates one of the key dehydrogenases called, one of the key reductases called nitrate reductase, and you see over here, and taxate is the major inhibitor of this reaction. So we also find it as the top metabolite. Also, this reaction, this nitrate reductase, has a protoheme and iron sulfur cluster in the core. And the second metabolite that we find is the protein. And also the substrate, the reduced product of this reaction is nitrate, and it's nitrite, and it's found also in our list. And finally, the downstream reductases also have some substrates and products. We're also able to cache those in our reporter list. So the results show that we can find the underlying biology of the transcriptional response for the knockout of this gene. Next, I'm going to show you some results on autism spectrum disorder. So this analysis done on this data set published in 2011. We have 58 samples. We are using Ercument metabolic network. And although autism is not primarily a metabolic disorder, as considered primarily as a metabolic disorder, many metabolic symptoms have been reported in the literature. So it's really hard to go over one by one on each metabolite, but I'm going to give you an idea about the related pathways that we find. So the first pathway is the secretion pathway, which adds glycans onto the proteins as a post-translational modification, while those are secreted out of the cell. And in autism literature, 7 CNVs are associated with this pathway, as well as a DIA1R mutation. Second, we find the fatty acid synthesis pathway. Actually, we find 17 intermediates in this pathway, and this pathway is also associated with autism. The third one is glucuronidation, which is the detoxification of xenobiotics, and lower levels of this pathway also associated with autism in children. The fourth one is the carnitine metabolism. In our case, we are talking about the production of carnitine from lysine, and dysregulation in this pathway has been associated with non-dysmorphic autism, and the TMLHE mutation is also associated, as well as lower abnormalities in acid carnitine levels in this pathway. Finally, we found glycine cleavage pathway in this resulting set, and AMT mutation is also associated with autism literature. So, and the interesting thing about this slide is none of these findings in the literature was found in 2011 when this gene expression dataset was published. So, if we were to run this algorithm in 2011, these will be novel predictions which show the promise of the algorithm for possible drug target findings. Next, let me compare reporter algorithm with our results. First observation is reporter algorithm and MIRA finds different results set with Jacquard distance 0.78 for the delta AFNR dataset. And the second observation was difference in the reaction connectivity for the reporter metabolites. So in the paper we claim that original reporter algorithm has a bias towards hub metabolites. Then we had a correspondence with the author of the original algorithm, and he said using the background correction reduces this bias as described in Patel and Nielsen 2005. And then we tried, and so a random data set had average connection of 4. MIRA had 5 and array had 75. So after the background correction, we found that array had 22. So it decreased this bias. It's still higher, but I think it's not a bias. So we are going to correct this with the journal. Next, so I've shown you lots of results on E. coli and also in autism, but can we find those just purely by chance? So if we had a random data set, would we be able to find those findings again? So that's why we did a permutation test. We shuffled the labels of those samples and obtained 100 different data sets and ran our algorithm. Then we found a ranking for each of those data sets. We sorted them and plotted them. So here at the bottom you see 101 lines, and the red one being the result of our data set, of our algorithm on the original data set. So as you see, for it nice dominates and each and every random line behind, showing that the results we find are empirically significant. So the top figure is just a zoom in the first 50 metabolites. So we did the same thing for the reporter metabolite with the background correction. As you see, the red line is now the reporter algorithm. It's one of the top three lines, but still not as significant as MIRA. So let me conclude. So we propose MIRA, which uses a combinatorial and multivariate approach based on mutual information to find the reporter metabolites. So MIRA works well with small sample sizes, considers multiple associated genes at the same time, does not cancel out significant changes with non-significant ones, and finally it works well with the E. coli and autism datasets that we consider. So let me acknowledge Carnegie Mellon and Lane Center and Ray and Stephen Lane Fellowship, and also let me thank for the travel funding for Department of Energy, as well as my co-authors and comments by Kiran Patel. Thank you.
Questions, please.
A great talk. So do you think metabolite differences could be used as fingerprints or finger types for different sorts of autism disorders? Because your examples were, different metabolites in different, types of autism. Do you think these could be turned into fingerprints for different autism spectrum disorders?
You mean the findings that our findings?
Yes. That is, particular metabolite differences would be indicative of autism spectrum disorder, as opposed to recovering them.
I see. Yeah, of course there can. There are many works that are trying to find the metabolic differences between autism, between cases and controls in autism, and as well as maybe different types of autism within the spectrum.
But do you think MIRA is able to do this as well?
Yeah, of course, yeah. That's what I believe. There are also metabolic works, but I think MIRA is also a prominent source.
Thanks.